Thank you very much. Thanks for coming out. Really what I was, in thinking about today's talk, I was trying to think about what's important to me, what's been important to my research and teaching for the last several decades. And it really is this. Over a century of research has really led to something important about us, that in every organ, in every tissue, in every cell, and in every gene of our bodies, we contain artifacts of over 4 billion years of the history of life. That's a really remarkable fact, and it's a fact that's important for basic discovery in biomedical, in the field of biomedicine and geosciences, but it's also important because it has applications to understanding things that are important for human health. So what I would like to do today is to give you a sense of the way we know this history, how we go about uncovering it, and how we go about leveraging that history to understanding and bettering the human condition. And we're going to try to do that in half an hour, so stand by. Most of my research, as you heard, for the past several decades, is derived from this slide. I saw this slide as a graduate student when I was doing my PhD, and I was like fishing around for a, pardon the pun, for a PhD thesis. And I was looking around, and I was taking this seminar where the professor was going through the greatest hits in evolution, and like in the third week, he showed this slide. And this is what we knew about of the transition from life in water to life on land. This is what we knew like in the mid to late 1980s. You see a cartoon of a fossil fish on top, critter from about 390 million years ago. And you have an early limbed animal from about 365 million years ago. That's, again, a cartoon of a fossil on the bottom. And I remember looking at this slide thinking, wow, that is a first-class scientific problem. How did critters evolve to land? How did those changes happen? Little did I realize at the time that by uncovering this transition, I was going to understand something about us, our basic biology, and things that have actual biomedical relevance, which is what we're going to talk about today. So what I want to do is talk about how we know this transition, and then I want to leverage that knowledge to the human condition. So I was training as a paleontologist at the time. I sort of shifted later to become more of a molecular biologist, but at the time I was trying to think about how I can find fossils that bridge this gap between a fish with fins on top and a limbed animal with limbs on the bottom, and to find maybe something that has say a flat head like a early limbed animal with a fin with arm bones inside. How do you find one of those transitional critters? So I did what paleontologists have done for over a century. And that is, if you want to find, say, a transitional fossil, say between fish and limbed animals, what you do is you look for places in the world that have three things. You look for places in the world that have rocks of the right age to answer your question. Remember I told you the fossil on top is about 390, the fish on top is 390, the limbed animal on the bottom is about 365. What you want is something in that time interval, which is the late Devonian. So rocks of the right age. The good news is geological surveys of countries around the world publish maps with their maps of what rocks are exposed, so you have access to those data. The next thing you look for are places in the world that have rocks of the right type to hold fossils. It seems trivial, but it's really important, and this is the hardest variable. Not every kind of rock holds fossils, because maybe the rock formed in the wrong environment, or maybe the rock formed in the right environment that the creatures lived in, but was subsequently deformed. Okay? You begin to learn that by studying the geological record. Finally, it does me no good if my wonderful rocks, the right age and the right type, are buried 12 miles underground or are in a politically inaccessible or dangerous landscape. So what we need is access, okay? Those are it. Now, you can all run out and be my competitors now because you have the toolkit it takes. Rocks of the right age, rocks of the right type, and then rocks that are accessible. And soon it became clear that the rocks of the right type were those, and I studied with colleagues, were those that reflected ancient delta systems. So that's what we focused on. The landscapes of the world are continually changing, but the idea became to look at rocks that were in the Devonian age, between 390 and 365, that's the right age, and then of the right type, to look at rocks that were formed like in an Amazon Delta kind of environment. Why? Because in a delta system, what you have are ancient seas, estuaries, rivers, and streams. These were the areas that were most likely hold these kinds of creatures where they lived. The also good news to this is this kind of environment is easy to see in the rocks. It's a package of sediments that is very identifiable if you know your geology. Okay? So the hunt for us became look for rocks of Devonian age, look for rocks that had signatures of ancient delta systems, and look for those places in the world that were exposed. And that's literally it. When you do that, a big world becomes a whole lot smaller with a few candidate places to look. Turns out the best candidate place was in the Canadian Arctic. And you could see it here. These are the islands of northern Nunavut in Canada. You could see Ellesmere Island on the right there in green, and the western Arctic, Melville Island, and so forth. Encircled in red there are where Devonian age rocks of the red age, rocks that were formed in ancient delta systems in the Devonian period, where they're exposed across the Canadian Arctic. I remember the first time I saw this diagram, I thought, okay, there's a research program that's going to go on for a decade, and I was wrong. It went on for a decade and a half. And you could see these exposures extend all the way across the Canadian Arctic. So that's literally what we did. We sat down on these rocks and spent years looking for a fish, say a flat-headed fish with fins, with arm bones inside, to tell us how the transition happened. And this is all a way of knowing as an entree, so how do we know? So we started in 1999 in the western part of the Arctic, and that's what camp looks like. And literally what you see in the background are the Devonian age rocks. And what we do is we walk with geological maps and look to see what bone is weathering out, fossil bones are weathering out on the surface, because it does weather out in the Canadian Arctic. That's a big desert. Turns out we were in the wrong place that year. We were in the middle of an ancient sea. Where we were, if you go back to the cartoon, is we were in that blue, in the ancient seaway. What we needed to do in future years was to go upstream into the estuaries and into the rivers. And so we went back, we did our geology, and turns out to go upstream geologically in the Canadian Arctic meant going east. So then we shifted our site the next year, in 2000, to sites in southern Ellesmere Island. You can see it's a little more montane. Then we had to find another site. That was great. We started to find fish, but fish that lived in ancient streams, but they were bits and pieces, not whole skeletons. We needed a place where the geology was right to gradually preserve fossils. Well, we found doing a lot of reconnaissance, we found this valley here that had the right kinds of rocks, very fine-grained rocks. It had a signature of the kind of rock that would do this. And then we found a place where there was a layer with fossil skeletons preserved. And we removed those fossil skeletons, and the big discovery was this. Doesn't look like much, but that's it. What you see here, well, I hope you see, is this little V right there. You train your eye. I know you're thinking I'm utterly nuts, but those maroon rocks are Devonian age rocks, and there's bone inside that. What I was looking at when I saw that was an upside-down front end of a jaw of a flat-headed fish. And remember, I told you flathead was what we were looking for. We brought it back, and this is in 2005, to the laboratory, that specimen. We subsequently found 20 more of them. Removed the rock grain by grain. A preparator did that, and you can see what's coming out. You're looking at the top side of a skull. You could see 2 orbits there. Another 6 months go by. Boom, looks like you have a flat head with eyes on top. There's 2 shoulders coming out. Remember, what we're looking for was a transitional creature between fish and land-living animal, maybe a creature with flat-headed fish with fins, with arm bones inside. And this is the creature. Right here is a cast of the skull. What we first saw was this V right here. This is what is sticking out. So this is a cast of the skull. It's about 4 feet long. What's really interesting about this is it's a perfect transitional fossil in many ways. If I was to have the whole thing here in front of you, you'd see it has scales in its back and fins with thin webbing. And it has other fishy traits as well. The texture of the bones is very fishy. But like a limbed animal, it has paired nostrils. Okay? It had both lungs and gills. We know that from the insides. And if we crack open the fin, what does it have? It has an upper arm with a shoulder, has an elbow and a forearm, even parts of a wrist. And I'll show that to you in a second. I'll pass this around. Don't worry, it won't break. It's a cast. My kids, when they're 5, they're not break. And you break it, I'll glue it back together. Anyway, so if you look at this thing, if you look, here's a phylogenetic tree, an evolutionary tree of what's going on here. Take our skeleton of our arm. What do we have? Its arm has 3 regions to it. 1 bone, shown in red. 2 bones, shown in yellow. The technical names are there. And the wrists and digits, shown in blue. That 1 bone, 2 bone, wrist, digit area is seen all the way in the fossil record, all the way to our fossil fish, which shows the 1 bone, 2 bones, blue pattern, but all set with a creature with fins with fin rays. So what we have is something here that has features of both a limbed animal and features of both a fish found at the right time in the fossil record. So that's how we go about knowing. Now, what do we do with what we learn? That's what I want to show. So this story of the transition from life in water to life on land is not some esoteric event in evolutionary history. It's an event that's embedded, artifacts of which are embedded in our own biology and actually have impacts to our lives today. Because what I could show you is you can trace the limb bones of a creature like this thing, Tiktaalik rosea, all the way to humans. You could trace the humerus from Tiktaalik to amphibians, to reptiles, to other mammals, to people. You could trace the elbow from Tiktaalik to amphibians, to reptiles, to other mammals and people, and on and on and on. There's an unbroken line. We see for the first time in Tiktaalik a neck in the fossil record, a wrist in the fossil record. So I can use the evidence of the fossil record, and I'll show you the molecular record in a second, to make this case that whenever you bend your wrist and whenever you shake your head, you're using structures that arose 375 million years ago as our distant ancestors evolved to walk on land. Now, this itself is no esoteric knowledge because it too goes ever deeper because it leaves artifacts in the DNA record. It leaves artifacts in the embryos of creatures that are developing today. And so that's where I'm going to shift to show you the next phase and how we can apply this knowledge even more broadly. One of the most fundamental puzzles of biology is you and me, each of us, are sitting in this room and we're about 4 trillion cells. Okay? 4 trillion of our own cells. There's a microbiome of a lot more. But of our own cells, there's 4 trillion of them, all packed in the right place. All right? We hope. We began as a single cell. Okay? So bodybuilding in a developmental sense is how do you go from that single cell to 4 trillion cells all packed in the right way? Well, obviously, it's an orchestration of genes being turned on and off during development, tissues forming and interacting. That process of development holds so many basic clues to biology and the human condition. It also is something that's fundamentally part of evolution. So let me give you a couple examples. If you look at the head of a shark, shown on the left here, you're seeing the top head of a shark a few weeks after it's conceived. It's either a shark or a skate or a ray. All the cartilaginous fish have something like this. What you see on top are the paired primordia for the eyes, and you see a series of swellings in the so-called pharyngeal area, and they're color-coded there. Light blue, dark blue, green, and yellow, and so forth. Those swellings are filled with cells. And each swelling is separated from the next by a little cleft. Okay, in a shark, what happens? In a shark, the first swelling becomes the bones of the upper and lower jaw. The second swelling becomes the gill bones that lie behind that, and the others become successively other gill bones along that. So what you're seeing in sharks is those swellings and those cells, those derivatives of those cells, are forming the upper and lower jaw, as well as the gill apparatus. And that includes the muscles and nerves and bones that control all that stuff. Okay, these are the so-called famous pharyngeal arches. They also form the gill slits and so forth, and other structures as well. What happens in humans and other mammals? Well, that first swelling becomes a portion of our lower jaw and 2 bones in our middle ear. The second swelling becomes a little bone at the base of the throat called the hyoid, and 1 bone in the middle ear. And the other 2 become parts of our voice box, as well as the muscles and nerves and bones that control all that stuff. What this means is, in a developmental sense, and also in an evolutionary one, many of the muscles and nerves and bones I'm using to talk to you with right now, and many of the muscles and nerves and bones you're using to hear me with right now correspond to gill structures in sharks and fish. We know that from the development. We know that from the DNA that drives the development. I can compare the genes that are active in the different gill regions of sharks and see where they're active in the human head and pharyngeal area. Likewise, excuse me, we see it in the fossil record. That is, I could trace the evolution of some of these gill bones, like this gill bone here, to become an ear bone in humans. That is where evolution takes us, to those kinds of comparisons. And they are very, very fundamental. Because if you look at one of the most remarkable discoveries in this field in the last 2 decades is that the basic toolkit of genes that drive the development of these structures, as well as the architecture of the whole body, is deeply shared among all animals, all multicellular animals on the planet. If you look, say for instance, at the genes that drive the patterning of our vertebral skeleton, okay, from cervical, thoracic, and the different regions of the body, and where our limbs are in that, and where our ribs are. There are a number of genes that are turned on and off in the embryo. So you see a human on top, you see a human embryo on the bottom, that's actually derived from a mouse. It turns out versions of those same genes are active in the development of the architecture of a fly, of architecture of other kinds of arthropods, of diverse worms, and so forth. And what are those genes doing? They're driving the basic architecture of the body as well. So these facts, these fundamental facts, are very important because there's a deep conservation, a historical connection between flies, worms, fish, mice, and people. We see that in the DNA, we see that in their development, and we see it in the fossils. Now, how do we probe even deeper? Let me give you an example from my own work. I'm going to show you this slide on top. You can see there's a human arm on top. We talked about the 3 regions. There's in the middle, that's a Devonian limbed animal with fingers. That's from later in the Devonian, from Greenland. Tiktaalik rosea, the fin of the creature you're passing around, is on the bottom. And then here's a zebrafish here, which is a workhorse system in laboratories today. Okay, so you have a variety of fins. The creature on the bottom is an extant fish, okay? And if you look at that fin, it's kind of hard to compare it to Tiktaalik. It's kind of hard to compare it to tetrapod limbs. Why? Because it has 2 kinds of bones in it. One kind of bone here are the fin rays, okay? Those form most of the fin. When you look at a fish swimming and you see the webbing of the fin and the bones between them, it's usually in the fin rays. The base of the fin, shown in black there in the zebrafish on the bottom, those are bones that are formed in cartilage. Okay, and there's no obvious comparison or similarity between those black bones in zebrafish to those things like tetrapods, limbed animals, and Tiktaalik. So how do you compare them? Well, it's interesting here because in evolution, one of the big transitions is you have the origin of fingers, as you go from something like Tiktaalik to tetrapods, but you have the loss of the fin rays. Okay, so there's 2 different trends, 2 different kinds of bones, and all these comparisons we really can't make. Well, it turns out that those genes I showed you before in the previous slide that are active in the development of the body, they're also active in the formation of the limbs. And we can ask some really important questions about the DNA that's relevant to understanding our own limbs. Let me show you an example. What you see here is a mouse. Okay, on the right is a mouse limb that's been prepared. You can see the cartilages and bones are standing out in the limb. You can see it's been cleared a bit so you can see inside. You're seeing the skeleton inside. On the left is the limb bud of the mouse. Okay, so what happens in the mouse is, just like us, the limbs originally form by sticking out the side of the body, so-called limb bud, and then it generates pattern, okay? And you can see it has that little paddle there forming where it's shown in black. That paddle is where the digits and wrists are going to form. What we've done there is we've labeled a set of genes, they're so-called Hox genes, a number of them, to show that some of those genes are very specific to the wrists and digit area. So you see in the early embryo, let me just go back, sorry, I hit the wrong thing. You see in the early embryo, the Hox genes are labeled. You can see all the tissues that are showing where they have Hox gene activity on the left, and then that's the developing skeleton. We could trace those cells into the adult stages, and if you do that and let it go, what you see is shown in yellow here are the labeled cells that have Hox, that had that gene activity. We've traced them all the way to the adult. What you should see immediately is that those cells that are marked by that gene activity are very specific to the wrist and digit, right? So what you have here in this gene, in these genes, is a marker for wrists and digits. Cells that experience that activity of those genes become wrist and digit, okay, in this. Furthermore, when you make a mouse that's lacking those genes, so you use CRISPR-Cas gene editing or any number of tools, what do you get? You get a mouse limb that has no wrists or digits, although the other bones are normal. So what I'm telling you here is you can use molecular biology to identify markers for the wrists and digits, whole gene networks that are responsible for forming this part of our bodies. Okay, you can do this for other parts of our bodies as well. We're getting very good at that in embryology and molecular biology. From my perspective, I was asking the question, how did that come about? Do fish have those genes? And if so, what are the genes that are making wrists and digits? What are they doing in fish? Right? And if so, can we leverage some basic biology out of that? So let's zoom in there. So what we did is the same experiments on fish. You're going to see zebrafish in a second. We labeled those cells, right? You can see how the wrist and digit is marked. When you do it in fish, number one, those genes are there in fish. And number two, they're active in the fins. And number three, they're not just active anywhere in the fins. Boom, they're active in the fin rays, and very specifically in the fin rays. And when you do CRISPR-Cas genome editing and you remove these genes, I don't have the slide, but what you do is you make a fin without the fin rays. So the comparison we're getting from molecular biology is when you compare a fish to a human, making a limb doesn't involve that many new genes. There are some. But in reality, what you're doing is using old genes in new ways. That the genes that made the fin rays in fish, that whole compartment, that are involved in specifying that development, are patterning the region of the wrists and digits in mice. Now, for those of you who know something more about developmental biology, what we're doing is actually now tracing those cells, the cell fates. We're looking at, it turns out the same population of cells in fish that form the fin rays form the digits in mice. That's really remarkable. Because now that we know that, we can ask the question, what drives those different paths of development to form different kinds of skeletons? So here's a case where evolution and knowing something comparatively has set up a new way of thinking of how new sets of experiments that we could follow to understand how cells differentiate. The other area to apply that we've thought about applying fish work and understanding of the fish tetrapod transition and phylogeny to thinking about is regeneration. We should be very jealous of salamanders, okay? There's a lot of reasons why we should do it. Number one, some of them can flip their tongues the length of their body in milliseconds. But the other is captured here. On the top, you see a normal salamander limb. If you cut off the limb, and you can do it at any level, it repairs, and then what happens as it repairs? Just go all the way down, it regenerates. And it regenerates virtually fully. The same skeleton, virtually the same vasculature, virtually the same muscles and nerves. And it does this in a very stereotyped way. That is, if you cut the limb off, after 1 or 2 days, it heals. You get the closure of the wound, okay? Then you start to get a whole population of cells, and after about a week, that form at the terminal end, and that population of cells is what sort of differentiates to become the new regenerative limb, including all those different tissues. So that little sliver of cells at the terminal end of a salamander limb has the potential for muscle, bone, artery, nerve in the right pattern, okay? It's not just limbs that salamanders do this for. If you cut their hearts, you cut their spinal cord, it's hard to kill a salamander because everything regenerates. It's really remarkable. And so that potential is seen in diverse tissues. So what do salamanders have that we don't is a very relevant question for humans. But there's another thing. It's not just salamanders here. So if you look at fish fins, and if you look at fish along the evolutionary tree, particularly ones that are the closest relatives of creatures with limbs, if you cut off their fins, we found this accidentally in the lab, where we'd keep them too closely confined with one another and they'd bite each other's limbs off. A postdoc in my lab noticed, well, these critters were eating each other, biting their limbs off. Occasionally, they'd survive, and they'd regenerate. So there's one where he actually did it. This wasn't bit. He actually did the surgery. After a week, you get a wound healing in the middle there, and after about 14 days, you start to get a proto-fin coming back. He let the whole experiment go with multiple fins, and it turns out if you trace them, after a month, they start to regenerate the cartilage of their fin. After a month and a half, the cartilages gain pattern. After 3 months, the cartilages gain the full pattern, such that if you compare the wild-type fin of a normal, shown on top of the fish like this, and one at 6 months, it's doing exactly what salamanders do. It's regenerating completely. And by the way, these fish do it with their spinal cords and their nerves as well. It turns out that if you look at the evolution, put all this regenerative capacity on an evolutionary tree, you see something very profound. What you see is that most basal creatures have the potential to regenerate and their whole bodies. If you cut a planaria in half, it'll make 2 planaria. cut it in fours, it'll make 4 planaria. If you look at most fish, they can regenerate their fins completely. We lack that ability. So it's not that these creatures have gained the ability to regenerate. It's that in our lineage, somewhere in the origin of reptiles and mammals, the ability to regenerate has been lost. And it's been lost in a very precise way. That is, if you look at that diagram I showed you before of a regenerating salamander limb, it turns out we're really good at wound healing. We thank you, we know that. But the blastema formation, that bolus of cells that becomes the wrists and digits, those cells never form. We never form that. We never form a blastema in most of our tissue. So what is missing in humans that's doing that? There's a number of areas that I think they're important here. So if I show you creatures at the base of the tree, some critters like planaria and hydra and other worms can do whole body regeneration. As you move further up, amphibians and fish can regenerate hearts and spinal cords, but mammals like us cannot do this. So there's been a general, when we look at a phylogenetic tree, there's been a general decrease in regenerative capacity over time. So what you see here is an evolutionary phylogenetic tree. We are placental mammals. Our closest mammalian relatives are marsupials and monotremes. You can walk down the tree, a very simplified tree. If you map regenerative capacity on top of that, there's been a general decline in regenerative capacity to get to mammals. And that sets up all kinds of predictions. And so forth, if you map gene activity, populations of stem cells, and other features in there, what you find is we have lost a number of capabilities that existed primitively. And a part of that has been a trade-off, and we can talk about in the discussion, between having an immune system that's really efficient and having stem cell populations that can regenerate the whole limb. Anyway, so I wanted to give a sense of how we know evolution in the fossil record, how finding fossils is not all dumb luck. There's a lot of hard work and prediction that goes along with it. And then how we can leverage that, because we have deep history in our bodies. In the next month and a half, we have I'm part of the U.S. Antarctic program. We have an NSF grant, a 5-year NSF grant. We have our second trip to Antarctica coming in December through February. I'm leading another expedition. This one is our second one down there to Antarctica. We'll be visiting the dry valleys and nunataks of the Transantarctic Mountains, which hold Devonian age rocks. This part of the world has rocks of the right age, 380 million years old, rocks of the right type, formed in ancient delta systems. And since they are in Antarctica, if they're not covered with snow or ice, they are complete desert. This is the rock. It's like the Grand Canyon, only with glaciers. And this is, if you're a paleontologist, these are the kinds of exposures you go for. So we're starting all again to look for very primitive sorts of fish. So I wanted to close and I'd take questions, but I think, I wrote a book called Your Inner Fish, basically celebrating the fact that in our bodies, we contain historical artifacts. But those historical artifacts really have driven what we are and what we're capable of. And we can leverage that history to design experiments that take us from an understanding of fossils and the transformation we see in fossils, but also understanding our basic biology, written in DNA, written in cells, and written in our embryos. So you can always ask the question like, who cares about your inner fish or these things? And I could say, well, the Nobel Prize Committee in medicine or physiology cares a lot. Because if you look at the Nobel Prizes awarded to basic biomedical research, what have they gone to? People working on flies, people working on mice. 2 Nobel Prizes awarded to 5 people in the last 13 years have gone to folks working on Caenorhabditis elegans, tiny little worm the size of a comma on a piece of paper. Yet that worm is telling us about how our genes are turned off in health and disease and how they're programmed to die. So, I like to think that as we discover cures to everything that ails us, from Alzheimer's to different cancers, that the breakthroughs that will extend and enrich our lives will in some way be based on flies, worms, and in some cases, even fish. I can't imagine a more powerful or more beautiful statement on the importance of our evolutionary connection to the rest of life on our planet than that. Thank you.